Hello there, this is Dan, biology tutor, and I just wanted to go through the topic biological molecules with you. This video is really for students that I have supplied a set of notes, a set of my notes on the topics of biological molecules notes, and you should also have a set of questions which I've picked to deal with all areas of this topic. And these questions are designed to be most relevant questions. They're not always the most recent questions, but they're designed to, the booklet is designed to cover all the possible areas that you might be asked in this module 1 topic. The other thing you should have is a set of answers for the question booklet, which I will supply to you, and then you may also have a set of what I call short questions. So it's short questions on molecules, and we'll be using this in this video. I will try to go through the topic, through the notes and through the areas that you need to know about. I won't be going through the questions at this stage. I'll go through the questions with you in a Skype session, and that'll be at a later stage. Just to say at the start here for new AS students, that you're probably in school, you're probably using this textbook. And you may also be using this here revision textbook. Now, both those are very good. This one here is by Mr. Napier, and he's the chief examiner. So if you're only going to get one of these two textbooks, I would get this one. And it covers everything you need to know. So I'm basically going to work my way through my notes here, and you'll see, maybe think that my notes that they're radically different from what you've got in school, or what you've been given out by your teachers. What I'm going to say about the notes is that my notes are designed to cover the important parts of the topic, the parts that you need to know about, and I've accentuated those parts. And some bits and pieces I may have appear to have left out, and maybe bits that you've done in class, and you think, well, those bits should be in the notes as well. So, for example, from talking to a lot of students would be saying that the first thing their teachers maybe did on this topic was to talk about the structure of water molecules, and maybe spend quite a bit of time talking about the structure of water molecules. Well, I have to say to you straight away that you don't need to know about the structure of a water molecule. Essentially, really all you need on water molecules is that water molecules have hydrogen bonds, and later on in the course, in a different part of the course, that'll become relevant. So you don't really need to know about water molecules. So you'll find in my notes then that I don't spend much time talking about water. I actually relegate water to the very back page of this biological molecules booklet, because it's really not that relevant. And you won't get any questions about the structure of water molecules, and you can search through past paper questions as many as you want. You'll just simply not find any questions about water. So so you forget about that. And the other thing you probably did near the very start of the course was to discuss the role of different maybe ions and atoms in biologically important compounds, like the role of iron, for example, in hemoglobin, and magnesium in chlorophyll, calcium in cell walls, that sort of thing, nitrogen in making proteins and so on. Most of that you can leave until later in the course. That it's a bit early to do that at the very start of the course. So I really put that on the back page of the notes there, and you'll see what I have there is sufficient. And in fact, if you go looking through past paper questions, you'll not ever see anything asked about that. I don't think I've only ever seen one question asked on that in 10 years, and it was just something along the lines of name a biologically important molecule that contains magnesium or something like that, and the answer was chlorophyll. So that's really not that relevant. Where this where this topic really starts is with is looking at carbohydrates, lipids or fats, proteins, and nucleic acids, the four important biological molecules that you need to know about. And it's quite detailed. Coming from GCSE, you maybe don't expect so much detail, but there's an awful amount of detail about the structure of these molecules. So you'll see on the front page of my notes, that basically lists the four types of molecules: carbohydrates, lipids, proteins, and nucleic acids. Teachers often neglect to say who the nucleic acids are. Nucleic acids are DNA and RNA, and we'll discuss those in some detail later on. DNA and RNA, those are nucleic acids. DNA actually stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. And we'll look at those. And you'll see on the front page of the notes there, the first thing I always do is include a table that compares the elements present in the four types of molecules. And you'll see there, just look at that table, that all four types of molecules, carbohydrates, lipids, proteins, and nucleic acids, all contain carbon, hydrogen, and oxygen. In lipids, you may get a phosphorus present as well, but that's only present in lipids called phospholipids. And you'll notice that proteins contain nitrogen, and that's very important. Proteins contain nitrogen, and sometimes they contain sulfur. Usually they contain sulfur as well. And nucleic acids, that's DNA and RNA, they contain phosphorus and nitrogen as well as carbon, hydrogen, and oxygen. Now, that wee table is not that relevant at this point, but you may come across a question, and a past paper question, and I've come across a question in the past where they presented you with information about mystery molecules. So they might say, for example, molecule A contains carbon, hydrogen, oxygen, but it also contains nitrogen. And then molecule B contains carbon, hydrogen, oxygen, nitrogen, and phosphorus. And so they're kind of give you a kind of a clue kind of thing, and you have to determine what the molecules are based just on the carbon, hydrogen, oxygen, nitrogen thing. And so you may come across a question where they're asking you to compare the different molecules, or maybe asking you to identify molecules A, B, C, and D, and so on. So that's what that's there for. just to, and you can see the pattern. They all contain carbon, hydrogen, oxygen, and it's just to learn that pattern then as to which who contains nitrogen and sulfur and so on. So, okay, so when we're dealing with the biological molecules, then you'll see some that you sort of recognized at GCSE. At GCSE, you would have just done carbohydrates, fats, and proteins. You probably wouldn't have mentioned the word nucleic acids, but you were aware at GCSE of DNA. Now, what you'll see with all of these molecules is that there are big molecules and small molecules. So big molecules and small molecules. And at GCSE, you would have recognized that a big molecule was a protein, and you could break a protein down into its small molecules, which are amino acids. So big molecule and small molecule. Big molecules, for example, starch, and you could break a starch down into smaller molecules, which were glucoses. And at GCSE, you would have known that if you broke down a lipid or a fat, you got fatty acids and glycerol. So you had big molecules and small molecules. In biology, various words are used for these big molecules and small molecules, and sometimes there's too many words and you get a bit confused with them. And for example, the big molecules are often called polymers or macromolecules. Macromolecules, maybe not a term you've come across, but that word was used in a past paper question. What is a macromolecule? Macromolecules. And the small molecules then are often called subunits or monomers. So for example, then a protein would be called a macromolecule or a polymer, and the small molecules then, the amino acids, would be referred to as the subunits or monomers of the protein. Okay, so it's those two terms. So there we are. Let's move on quickly to carbohydrates. Carbohydrates all contain carbon, hydrogen, and oxygen. So carbohydrates are a family of molecules. You're aware of carbohydrates from GCSE. At GCSE, you'd know that carbohydrates are good for energy, and you'd associate carbohydrates with things like potatoes and bread and such foods. Flour and those sorts of things contain a lot of carbohydrates, and you would have the general idea that carbohydrates were good for you. They supplied energy and so on, but that if you had too much carbohydrates, too much food, that too many carbohydrates weren't good for you either. So a certain amount of carbohydrates. So consider carbohydrates to be a good food molecule. Well, we see here from the notes, in the carbohydrate family, as it were, is made up of three different kinds of molecules, or three types of molecules. You've got your monosaccharides, and those are new words to you. You haven't come across the word saccharide before. Monosaccharides, disaccharides, and polysaccharides. The monosaccharides are the single unit molecules, single monomers, just little molecules, such as glucose and fructose. And the polysaccharides are starch, glycogen, and cellulose. So polysaccharides are the big molecules, starch, glycogen, and cellulose, and the little molecules, the monosaccharides, are glucose and fructose. In between, you have things called disaccharides, and really disaccharides, as the name says, di means two, so disaccharides are just two monosaccharides joined together. And you only have to know two disaccharides. You have to know maltose. So maltose is made up of two alpha glucoses, as they're called, alpha glucose, and I'll come back to the alphabet in a minute too. So maltose is two alpha glucoses joined together, and the other one is sucrose, and sucrose is a alpha glucose joined to a fructose. Look at the pattern there. Maltose, two alpha glucoses, sucrose, an alpha glucose, and a fructose. Okay, those are the only two you have to know. Sometimes schools will also include lactose, L-A-C-T-O-S-E, a disaccharide made of glucose and galactose. Not actually on the specification, but sometimes it's included in school notes. So monosaccharides and disaccharides and polysaccharides. So what about so you have to know those. You have to know those. Then you have to know the two monosaccharides, glucose and fructose, the two disaccharides, sucrose and maltose, and you have to know the three polysaccharides, starch, glycogen, and cellulose. So that's quite a lot of molecules to begin with. So the carbohydrate is quite a complicated family, and you sort of think, well, I have to know all of those. That's 2 and 2 is 4, and that's 7 different things. But it's actually not too difficult. It's not too difficult. First thing is glucose and fructose. Well, glucose and fructose, when you draw them and in your notes and in the textbook, they look quite complicated. And they've got lots of carbons and hydrogen and oxygens. And you think, oh, do I actually have to learn this? Well, you'll see in the bottom of page 2 in my notes there, I have a picture of what's called alpha glucose. And you can see that it's basically got a hexagonal shape. So it's got a 6-sided shape, so there's like 6 corners to it. Hexagonal shape. And you've got carbons and oxygens and hydrogens and all, and that's alpha glucose. Do you have to learn that off by heart? Absolutely not. No. You'll never be asked the structure of glucose. You won't be asked to draw it. You won't be asked to put in hydrogens or any of that sort of thing. That's just not what you're going to be asked. What do you need to know about it? You need to know that its formula is C6H12O6, and you learned that at GCSE. Glucose is C6H12O6, so we've got 6 carbons, 12 hydrogens, and 6 oxygens. C6H12O6. And because it's C6, it's got 6 carbons, it's referred to as a hexose, Hexose. A hexose is a 6-carbon sugar, or 6-carbon monosaccharide. Now, I slipped in a word there, and I sort of thought about it. I said it's a 6-carbon sugar. Sugar? Where does that word come in? Well, sugar is a word, really not a technical word, but here's what it means. The word sugar includes the 2 monosaccharides and the 2 disaccharides. So sugars, glucose, fructose, maltose, and sucrose. So they're referred to as sugars, and it's a sort of a loose scientific term. It's not actually a technical term, but the word sugar, it does slip into the vocabulary. So sugars, glucose and fructose, maltose and sucrose. Those are the 4 sugars as they are. So come back to what I was saying. What do you need to know about glucose? You know that it's got 6 carbons. It's a hexose, a 6-carbon sugar, a hexose. And I would know its shape. It's hexagonal in shape, 6 sides, hexagonal in shape, and it's that shape is well, the fact that it's got 6 carbons is important. And the only other monosaccharide you need to know about. Well, sorry, before I leave glucose. Glucose has got 2 forms. It's got there's 2 types of glucose. An alpha glucose and a beta glucose. Alpha glucose and beta glucose. Okay, how are they different? Well, you'll see in the notes there, you'll see that it's a very minor difference. There's a H and an OH group there in the glucose. If you switch those around, then with the H group up up at the top, you've got glucose. If you put the H group down at the bottom, put the OH group up, you've got beta glucose. Is this important? Are you asked to identify which is which? No, absolutely not. Do you need to know that there are two forms of glucose? Absolutely yes, alpha glucose and beta glucose. And later on you'll discover why that's important, alpha glucose and beta glucose. Fructose. You look at fructose there. Fructose looks the same as glucose. 1, 2, 3, 4. It's got 6 carbons again. So it's a 6 carbon molecule. This time you'll see it's just, in its shape, it's just 5-sided, but it's got 6 carbons. So it's also a hexose because it's got 6 carbons. Do you get asked to draw the structure of fructose? Absolutely not. You don't get asked at all. You don't get asked to draw the structure of any of these. Just be aware that they are both hexoses, Hexoses. They are both 6 carbon sugars. They're both of 6 carbons. Where is glucose important in the world? Glucose is made in photosynthesis by plants, and it's an energy store. It contains energy, contains energy, and you can get the energy out of it in a process called respiration. You can extract the energy from. Fructose is similar to sucrose. That's all I've said. Similar to glucose, 6 carbon. Now, there's a couple of other so-called monosaccharides that you need to know about, and they are deoxyribose and ribose, and they're both pentoses. They're both 5 carbon sugars. In other words, they both contain 5 carbons, and they're called deoxyribose and ribose. Why are they important? Well, it turns out that they're important in the structure of DNA. To make DNA, you need a deoxyribose sugar, as it's called. You need a deoxyribose. And to make RNA, I'm going to attempt to write something on the board here. So to make DNA, you need one of these deoxyribose 5 carbon sugars like that. And then deoxyribose also contains then a thing called a base, and it contains a thing called a phosphate group. So to make DNA, you need lots of these things. These things are called nucleotides, and each nucleotide contains one of these, which is a deoxyribose, a 5 carbon sugar. Okay, and DNA's got 2 strands to it. You maybe haven't done DNA, but if you're making RNA's just got 1 strand, single-stranded nucleic acid. We'll come back to it. It doesn't have deoxyribose. It's got R, which is ribose, the sugar ribose. So in RNA and DNA, the sugar is called deoxyribose. In RNA, it's called ribose. So these two 5 carbon sugars are important in another part of the course. They're important in the structure of DNA and RNA. Okay, so that's your monosaccharides. They're the basic units of other molecules. Now, the one that's most important here that we've done is glucose. And glucose, glucose then has 2 forms, alpha and beta glucose. So we're going to talk a bit about those later. Alpha and beta glucose. Okay, before we discuss alpha and beta glucose again later on, let's look at the disaccharides. Disaccharide means double sugar. That's actually what that term means, a double sugar. So it actually means 2 monosaccharides joined together. And you have to know 2 of these. You have to know maltose. Maltose is a disaccharide. It's made up of 2 monosaccharides stuck together. And the 2 monosaccharides that make up maltose are called 2 alpha glucoses. 2 alpha glucoses joined together to make a disaccharide maltose. And the other disaccharide that you need to know is sucrose. And to make sucrose, you join an alpha glucose and a fructose. So those are the 2 you need to know. How are these molecules joined together? They're joined together in what is called a condensation reaction. So 2 alpha glucoses are joined together in a so-called condensation reaction to make the disaccharide maltose. And the bond that is formed when the 2 glucoses are joined together is called a glycosidic bond. Glycosidic bond. In fact, glycosidic bonds are anywhere where you join glucoses together. Anywhere in carbohydrates, glycosidic bonds are the bonds of carbohydrates, joining 2 alpha glucoses together to make maltose, or alpha glucose and fructose to make sucrose. Join them together, you make a glycosidic bond. If you wanted to split them apart again, you want to split the two alpha glucoses apart, and you want to break, in other words, you want to break down the maltose, then it's called a hydrolysis reaction. A hydrolysis reaction. And you separate the two glucose molecules, two alpha glucose molecules. And you'll see then on page 4, I make a point there just saying monosaccharides and disaccharides are referred to as sugars. Okay. Just before we leave monosaccharides and disaccharides, going back to page 2 there, you'll see that underneath monosaccharides and disaccharides I've written the word soluble. So both of these are molecules that dissolve in, both these groups are molecules that dissolve in water. So glucose will dissolve in water, fructose will dissolve in water, sucrose will dissolve in water, and maltose will dissolve in water. All these things dissolve in liquid. Where do you find these? Well, you find them all around you. Glucose and fructose you would find a lot in energy drinks. Fructose comes from fruit. You find a lot of it in energy drinks. Maltose you would find when seeds are germinating and breaking down. You won't be asked about that. And sucrose is the one sugar that you're familiar with, everybody's familiar with, because it's the sugar you put in your tea or coffee, or you put in your cereal. That's sucrose. Sucrose. Okay, on we go. So moving on then, I'm moving fairly fast here because there's quite a lot to cover in this topic. So I'm moving on then to polysaccharides. So here are polysaccharides. Well, there's 3 you have to know. You have to know starch, glycogen, and cellulose. How are these similar or different to each other? Well, let's say that starch and glycogen are both energy storage polysaccharides. Starch and glycogen are. Whereas cellulose is not an energy storage molecule. Cellulose is a structural molecule. In other words, come back again, starch is found inside plant cells, where it acts as an energy store. Starch is stored in plant cells. It acts as an energy. How does the starch get inside plant cells? Well, during photosynthesis, the plant cell makes glucose, if you remember from GCSE. So during photosynthesis, glucose is made, and then the glucoses are joined together to create starch, and that's stored inside the plant cell for later, as it were, to provide a source of energy later on. Inside our cells, we have a different energy storage molecule called glycogen, and you find glycogen mostly inside our muscles and inside liver cells, and it's an energy storage molecule as well. So those 2 are energy storage molecules, whereas cellulose is very different. Cellulose doesn't provide energy. It acts as a structural carbohydrate, and you find it in the cell walls of plant cells. You find it in the cell walls of plant cells. So we're going to look at those 3. Starch and energy storage molecule found in plant cells. Now, starch is a bit difficult because it's a bit awkward, because it turns out that starch is actually made up of 2 molecules. 2 molecules. The reason this happened is that starch was discovered a couple of 100 years ago, and at that point science just said, okay, this stuff, we're going to call it starch. But then more recently we've discovered it's more complicated. With better scientific techniques they've discovered, oh, starch is not just 1 molecule, it's made up of 2 molecules. Oh, and the 2 molecules have long, strange names: amylose, AMYLOSE, and amylopectin. 2 molecules present in starch. How are they similar? Well, the first thing to know is both are made of alpha glucoses. Alpha glucose is the monomer or the subunit of starch, of amylose and amylopectin. Both are made of alpha glucoses, and the alpha glucoses are joined together with glycosidic bonds by condensation reactions. How are these 2 molecules different? Well, amylose is a long straight chain molecule made of chains of glucoses joined one to the next, and you'll see in the diagram there on page 5, you'll see it's a long chain molecule, and it's wound into, the long chain is wound into a kind of a spiral shape. Okay, whereas amylopectin is, the alpha glucoses are joined in such a way that you've got something like a sort of branch structure. So kind of like branches of a tree, bits sticking out with glucoses sticking out. So one is long, one is long slender molecule, and the other is a branched molecule. But both of them are basically two molecules found in starch, and both of them are for energy. How do you get energy from these molecules? You have to take off a glucose. You take off the individual glucoses, and then you use the glucose in respiration. And you remember respiration from GCSE, aerobic respiration we're talking about here. Aerobic respiration will be glucose plus oxygen, and it gives carbon dioxide and water and energy. And aerobic respiration releases a lot of energy from the glucose. You get a lot of energy. So to get the energy from these molecules, you have to remove the glucoses, take off the glucoses. So you can remove the glucoses then by hydrolysis reactions. If you want to remove a glucose, you have to disconnect it from the glucose beside it by a hydrolysis reaction. So those are amylose and amylopectin. The third molecule there then over the page, on page 6, is glycogen. And you can see glycogen is also, it's an energy storage molecule in our bodies, and it's also branched. It's made of alpha glucose, but it's a branched molecule. So in a way, it's quite similar, if you turn back then, it's quite similar to amylopectin. So glycogen and amylopectin are both branched molecules. Now, sometimes you get asked a question, but what's the advantage to having these big molecules, as it were? What's the advantage? Well, for example, whenever a leaf makes glucose, why does the leaf then take its glucoses and join them together to create starch, or amylose from amylopectin? What's the advantage of joining all the glucoses together? And you'll see there on the bottom of page 6, there are some suggestions as to what the advantage of creating these big molecules. And they're described as good energy storage, good ways of storing the glucose. They're good ways of storing glucose, and because they are compact. That's the first thing. So what do I mean they're compact? Well, if, for example, a leaf cell has made lots of glucose molecules in photosynthesis, it's a good thing if those glucose molecules, instead of letting them all float about inside the cell, if they're all sort of stuck together, that creates they take up less space, and the whole thing's more compact. So by sticking them all together into this molecule, into amylose or amylopectin, you make a very compact molecule, takes up less space. You can easily remove the glucoses whenever you need them. They're easily hydrolyzed, it says there. The glucoses can be quickly released, so you can quickly remove the glucoses again when you need them for respiration. Whenever you stick all the glucose together into amylose or amylopectin, you go from a molecule that's soluble. The glucoses are soluble in the cytoplasm, in the water in the cytoplasm, whereas the starch is insoluble. And that means it's less likely to dissolve out of the cytoplasm, and it's less likely to have any osmotic effect. So we'll come to that later, that's a different part of the course, but essentially you've created starch, which are starch or glycogen or any of these molecules. They are insoluble and they don't have any osmotic effect on the body because they're insoluble. Now here's a classic question that's asked then about these three molecules, amylose, amylopectin, and glycogen. And the question you'll see at the top of page 7, and this is a standard question, that's why I put this question straight into the notes. So why are amylopectin and glycogen more readily available sources of energy than amylose? In other words, why is it that amylopectin and glycogen will provide you energy more quickly than amylose? And the answer is to do with the fact that amylopectin and glycogen are branched. They're branched, and because they're branched, there's more terminal glucoses. And what do I mean by terminal glucoses? I mean there's more glucoses at the ends. More glucoses at the ends. So you've got a lot of branches, then you've got a lot of glucose down at the very ends. Whereas if you look at amylose, because it's a long straight chain molecule, you've only got a glucose at this end and a glucose at this end. The significance of that is that the glucoses are taken off one by one. And so you take off an amylose, you would take this glucose off, and you couldn't take off the second one until you take the first one off. And so on. So you have to take the glucoses off by one, Whereas with amylopectin, because it's branched, you can be taking off glucoses all over the place because there's lots of glucoses at the end. So you've got more. And the way this is often put in your notes or in the answer scheme is that you say that amylopectin and glycogen are branched. And because they're branched, then there's more so-called terminal glucoses at the end. And if there's more terminal glucose, then it makes the molecule more easily hydrolyzed, broken down, as it were, to release these glucoses. So you can get the glucoses more quickly. And if you can get the glucoses taken off more quickly, then you can get more energy released more quickly in respiration. That seems like an odd kind of question, but that's a standard question. Watch out for that. And now you'll see that I've added an extra wee note there with an asterisk. I've said glycogen is more branched than amylopectin. And that means that glycogen is more readily, provides energy more quickly. It's more easily hydrolyzed and more quickly. It provides glucoses more quickly than amylopectin because it's more branched. So the more branching, the better for speedy supply of glucoses for respiration, for speedy supply of energy to the cell. So that's those. Now, sometime later, I'll either in a Skype session or maybe in another video, I'll go through all the questions, through the question booklet, and discuss the answers to all the questions. But at this stage, I'm just attempting to go through the notes fairly quickly and just maybe make a bit clear some of the points there. And if you have questions at this stage, you can ask me in a Skype session. We can discuss your questions. The last carbohydrate here, and that's is cellulose. And you see, the first thing you know about cellulose, then it's a structural carbohydrate. It's not for energy. It's not about supplying glucose for energy. It's not an energy source. It's a structural carbohydrate. It's found in cell walls. Cellulose molecules consist of straight chains of beta glucoses. And here you meet the importance of beta glucose. When you go back to starch and glycogen, that was all alpha glucoses. So they were all made of alpha glucoses. Starch, that's amylose, amylopectin, and glycogen, were all made of alpha glucoses, whereas cellulose is beta glucose. So you associate beta glucose with cellulose. Beta glucose, cellulose, beta glucose, cellulose. And that's really the only time you're going to come across beta glucose. It's the only time you're going to come across this alpha and beta glucose thing at all. It's just knowing that cellulose is made up of beta glucoses joined together, whereas amylose and amylopectin and glycogen are made up of alpha glucoses joined together. And that's only significance. You're not ever going to be asked to draw the structure of alpha glucose or beta glucose or say what the difference between them is. Turns out there's something called structural isomers. Something from chemistry, structural isomers, but you don't need to know that. Okay, cellulose then made up of beta glucoses. And if you can see from the notes there, the beta glucoses are joined in long straight chains. Long straight chains of beta glucoses. And something that doesn't come up too clearly in the notes there is that between the chains, and I've just kind of highlighted it there, between the individual chains, there are what are called, I've said there are hydrogen bonds. So there's hydrogen bonds between that chain and the chain below. So each chain is hydrogen bonded to the chain beside it. Sometimes the hydrogen bonds are referred to as cross-links. And the cross-links are important in making cellulose have high tensile strength. High tensile strength. Cellulose is a very strong material. To stretch it, it's hard to stretch it. And part of its strength is provided by those cross-links. What if you come across cellulose having a high ability to withstand stretching? Well, you did at GCSE. You learned at GCSE that plant cells wouldn't burst whenever they filled up with water. They expanded and became turgid, but they didn't burst. And why did they not burst? They didn't burst because their cell wall on the outside of the cell prevented them bursting. So the cell wall acts as a kind of a tough outer coat on the plant cell that won't stretch too much. And that's to do with, that's called stretching strength. It's called tensile strength. So the standard question that's often asked is that what gives cellulose its high tensile strength? So you see that question at the bottom of page 7. What gives cellulose its high tensile strength? And you would say that the marks for that would be beta glucoses are bonded together in long straight chains. Long straight chains, so beta glucose is bonded together, And then the second point is to say that there are hydrogen bonds, also known as cross-links, between the individual chains, and that adds extra strength to it. So it adds what's called tensile strength. There's a third point that's saying that the cellulose molecules are created into what are called microfibrils, and the microfibrils are sort of bunched together as well to create a stronger molecule. Just to show you something for a second, something that you come across that's made of cellulose is paper. And in paper, you see here I'm tearing paper, and when paper is made of cellulose, and if you look closely, you can do this yourself. If you tear a piece of paper, you'll see like wee fibers sticking out of the ends there. Those are the cellulose fibers that make up the paper, and that gives the paper the paper, so cellulose is quite strong, so you can stretch paper. It's very strong. Obviously, there's a lot of cellulose in paper. A cell wall wouldn't be as thick as this sheet of paper, but it contains cellulose, and cellulose has what's called high tensile strength and high stretch strength. So that's about as much as you'll be asked about cellulose. Later on, I'll look at questions on cellulose. So I'm turning the page then just to look at. This is a summary table then to compare the carbohydrates. Compare the polysaccharides actually. So it's amylose, amylopectin, glycogen, and cellulose, and it's just saying it says in each case what the monomer is. That's the small molecule that makes them up. So amylose, amylopectin, and glycogen, each made of alpha glucose, and cellulose, beta glucose. Their shape, I'll give you a little bit of their shape there, and where they're found. Amylose and amylopectin are starch, and where do you find starch? You find starch inside plant cells. And you actually find it inside chloroplast. And what's its function? What's the function of starch? Energy stored in plant cells. Energy storage. Now it's important to say energy store. I mark exams, and sometimes students will just say it's a storage molecule. Starch is a storage molecule. Now that's a bit vague. What does it store? Heat, light, sound? You need to say energy storage. So it stores energy. So it's a molecule that has a store of energy. So I accentuated the word energy there just a minute. And glycogen is an energy storage molecule in our bodies, inside our cells. And cellulose then is a structural molecule. So it's in plant cell walls where it's a structural support. By the way, in these notes, I attempt to put in the very important stuff, the important material, and focus in on what needs to be asked. Because I've been marking exams for such a long time, I'm familiar with the sorts of things that are important and the things that are maybe less important. So some things I don't put here at all because they never get asked. Okay. Food tests. You may or may not have done talked about food tests in class. I'm sure you haven't actually been allowed to do your own food test. The teacher maybe has done a food test. You'll see that just to finish, keep working on carbohydrates here. I want to go on and look at lipids and proteins, but I think that we should talk about the food test for carbohydrates. Well, if you list all the carbohydrates, then you have glucose and fructose, maltose and sucrose, and then you have glycogen, you have starch, glycogen, and cellulose. And you think about food tests for all of those. Well, first of all, let's talk about the monosaccharides and disaccharides. So the monosaccharides and disaccharides. Monosaccharides, glucose and fructose. Disaccharides, maltose and sucrose. Monosaccharides and disaccharides. And as I said before, those monosaccharides and disaccharides are often referred to as sugars. It's not a very technical word, but the word sugars is used. Sugars. So when you say sugars, you mean a monosaccharide and a disaccharide. Now, if I went back to GCSE, and I said, what's a food test for sugars? How could you test for sugar present in food? And you probably did that in GCSE. You probably brought in bits of biscuits and sweets and peanuts and chocolate and stuff, and you test them all. And you test them with Benedict's. Benedict's solution, or Benedict's reagent as it's called. Basically, you mix the food with Benedict's, and then what did you do? You heated the food, usually in a test tube. Heated it, and you got a colour change of blue to brick red. Blue to brick red, and that indicated that the food contained sugar. Now, the four sugars at AS level then are glucose, fructose, maltose, and sucrose, and you would expect to be able to test those with Benedict's and get a blue to brick red colour to indicate that they're present. Now, it turns out that the first three, glucose, fructose and maltose, they react with Benedicts. You just heat them with Benedicts and you get a blue to brick red colour. Okay. Now, it turns out that glucose has its own specific test. It is a test called Clinisticks. And Clinisticks is a precise test for glucose. Clinisticks won't respond to maltose or fructose or any of the others. So it's precise for glucose. But Benedict's test will work for glucose, fructose and maltose. But here's the thing. The Benedict's test will not work for sucrose. It won't work for sucrose. Glucose, fructose and maltose are referred to as reducing sugars. That's a strange term, reducing sugars. It's basically because those three sugars reduce the copper ions in Benedicts. You don't need to know that, but that's why they're called reducing sugars. So those three react with Benedicts directly. You have to heat them with Benedicts and you get a blue to brick red colour. But sucrose won't. However, there's a way that you can find out that sucrose is present, and there's like a 4 or 5 steps in showing that a solution contains sucrose. So, for example, if I had some sucrose in this bottle of water, you would have to do the following test to prove that there was sucrose present here. You'd have to take some of the water and first of all boil it with hydrochloric acid. So you boil some of the liquid that contains the sucrose with hydrochloric acid. You'll see the list of points there. I've said sucrose is called a non-reducing sugar. I've said sucrose won't react with Benedicts directly, won't react with Benedicts. It has to be heated with hydrochloric acid. So you have to heat the sucrose solution with hydrochloric acid using a water bath. Water bath's a safety thing. Then neutralizes sodium hydroxide. Neutralize the solution with sodium hydroxide. And now you can add Benedicts and heat, and it will react. But you have to go through the hydrochloric acid thing first of all. And basically, hydrochloric acid breaks the sucrose down into what? What does it break the sucrose down into? It breaks it down into its monomers, alpha glucose and fructose. And once you've got those two then in the solution, then you can put in some Benedicts and they'll react with the Benedicts. So, so sucrose is a bit awkward. And it won't react directly with Benedicts. You have to put it through this heating process with hydrochloric acid. Who also got a starch test? Well, we know the starch test from GCSE. Starch will react with iodine and give a blue-black colour. Glycogen and cellulose. There's no test for those, so you don't have to know it. So really, you just have to know the test for starch and the test for the 4 sugars. Okay, so that there would be asked about then in the practical exam, the practical written exam. Okay. On we go to, just check on our time. Okay. On we go to lipids. So lipids, also known as fats, also known as triglycerides. Triglycerides. And that's something that's caught students out in the past. In the past, students have been asked, what's the role of triglycerides? Give some roles for triglycerides. And they thought, do we do triglycerides in class? Oh, don't recognize the word triglyceride. Actually, it's just an alternative name for a lipid, or as is the word fat. So if I said to you from GCSE even, I said, give a function or a role for fats or lipids, you would say, oh, fats, energy store, insulation around your body, so you have fat all around your body, insulates and keeps you warm, and protection. You've heard also that fat's present around your heart and around your vital organs to sort of stop them being knocked about, sort of cushion. So those are 3 things, three functions of triglycerides. They may ask you, they may just say triglyceride, and you think, oh, triglyceride, they mean fats, they mean lipids. Okay, we'll come back to that. Plant lipids are often called oils. So we talk about oils, vegetable oils like peanut oil or olive oil. And then animal lipids are often just referred to as fats. So one that you wouldn't come across very much, lard, L-A-R-D, which is just animal fat. Okay. Other examples of lipids are phospholipids and cholesterol. You've heard of cholesterol. Cholesterol is a kind of a lipid that's in your bloodstream that has something to do with blocking up your blood vessels. We'll have a look at that later. Not, you don't have to know that in the course. Right? And then phospholipids, it's to do with cell membranes. Cell membranes are made of phospholipids. Strange idea. Cell membranes are made of fat. Yeah. Turns out, I mean, at GCSE, here's what a cell membrane looks like at GCSE. It's just a line. But when you look at it in some detail, yes, level, you discover it's not just a line. It's got a very complicated structure, and it's made up of, among other things, phospholipids, lipids, and it's also made up of protein. So we'll see that later. I said their fats and oils are called triglycerides. Why are they called triglycerides? Well, triglycerides because it's this thing about there's one glycerol molecule and 3 fatty acids. So one glycerol molecule is joined to 3 fatty acids. Now, you don't need to know the structure of glycerol, and you don't really need to know the structure of the fatty acids. And we'll come to what you do need to know in a minute. But you need to be aware that to make a triglyceride, you need to start with 1 glycerol, and then you need 3 fatty acids. And you join the 1 glycerol to the 3 fatty acids. What kind of reaction is involved in joining these together? The answer, once again, is a condensation reaction. So joining each fatty acid onto the glycerol is a condensation reaction. So there's actually 3 condensation reactions because you have to join 3 fatty acids on, 3 condensation reactions. And you create this thing then called a lipid or a triglyceride. The bond that's formed in these condensation reactions, the bond between the glycerol and the 3 fatty acids, is called an ester bond, E-S-T-E-R, ester bond. And in fact, you have 3 ester bonds because you have a bond between each of the fatty acids and the glycerol. So you have 3 bonds, 3 ester bonds. 3 condensation reactions, 3 ester bonds formed. And of course, if you wanted to break apart the lipid, then you would do hydrolysis reactions. Just talking about condensation and hydrolysis reactions, you've come across hydrolysis reactions at GCSE. Hydrolysis reactions are what happen during digestion, aren't they? They're what happens when you're digesting your food. Because you eat, for example, you eat a protein in the meat or whatever you eat, and the protein goes down into your stomach and digestive system, and it gets digested. And really gets broken down into amino acids. So it's really what happens in your stomach when the protein's been broken down is hydrolysis reactions. Same with you eat some lipid, eat some fat, it gets hydrolyzed or broken down in your digestive system into fatty acids and glycerol. Same with eat some starch, it gets broken down into alpha glucose. So hydrolysis reactions are what happen during digestion of your food. Okay. There's a point there just at the top of page 10, which is important for a question that once was asked. When you do break down a triglyceride, you get glycerol and the 3 fatty acids. It turns out that the fatty acids, because they're acids, they have a low pH, so they create a low pH. So you get a more acidic condition. You may come across that question again. Now, and the next bit then is just about fatty acids, different types of fatty acids. And really there are 2 types of fatty acids that you have to be aware of. There's a number of different fatty acids, but there are 2 types. There are saturated and unsaturated fatty acids. Saturated and unsaturated fatty acids. And you might have heard those words in the media to do with good fatty acids and bad fatty acids. You would generally have learned that it's better to eat foods that contain unsaturated fatty acids rather than saturated fatty acids. When you associate unsaturated fatty acids with plant oils, whereas you would associate saturated fatty acids with animal fats. And so you would generally an idea that eating plant foods, plant oils is better than eating animal fat. What's the difference between saturated and unsaturated fatty acids? Well, a single line gives the difference. Unsaturated fatty acids don't contain any double bonds between the carbons. Unsaturated fatty, sorry I got that wrong, right? unsaturated fatty acids contain double bonds between one or more double bonds between the carbon atoms, whereas saturated fatty acid has no carbon-carbon double bonds. Note both of them have a carbon double bond at the end, C double bond O at the end, but that doesn't count. It's double bonds between the carbon atoms. So you see, an unsaturated fatty acid has some carbon-carbon double bonds, whereas saturated fatty acids don't. Functions then, just run on down, functions of triglycerides, already said energy store, protection, and insulation, those three. Phospholipids, mentioned at the bottom there, are special lipids that are involved in making the cell membrane. Okay, so turn the page there, you sort of see a picture of a phospholipid. Phospholipid looks like a lipid, except it's missing a fatty acid, so it's got one less fatty acid. So a phospholipid has glycerol and just 2 fatty acids. And the missing fatty acid has been replaced by a phosphate group. So in a phospholipid, one of the fatty acids has been removed and replaced by a phosphate group. And often it'll be drawn, often this phospholipid, well, one end of it, the end with the phosphate group is often described as the head end of the molecule, and the 2 fatty acids that are sticking out are referred to as the tails of the molecule. And sometimes the molecule is just drawn as if it has a head and 2 tails, as it were. So a head and 2 tails, so often it's drawn, the phospholipid molecule is drawn like that. This is the head end. This contains the glycerol and the phosphate, and these are the 2 fatty acids. And the reason it's drawn like that is kind of a shorthand. It's drawn like that because we're going to show where it's involved. It's involved in the cell membrane. And here's some more new words for you. This end here, which is called the head end, is described as being hydrophilic. Hydrophilic, whereas these tail ends are described as being hydrophobic. Hydrophilic means attracted to water. Hydrophobic means repel from water. And if I could explain that maybe with a wee diagram here. Imagine you had a beaker of water. Say you had a beaker of water and you put in some phospholipids into it. This is the beaker. You put some phospholipids into the water. Here's what you would expect the phospholipids to do. You would expect them to stand on their heads like that, with their, as it were, their tails in the air, because their heads are quite happy to be in the water because they're hydrophilic, but the tails want to get away from the water because they're hydrophobic. So this is a molecule that has one end that's quite happy to be in contact with water, and the other end wants to get away from water. And these molecules, these phospholipids, are very important in the structure of the cell membrane. And you can see the wee diagram that sort of shows how they're arranged in the cell membrane. They're arranged actually in 2 layers. The cell membrane contains 2 layers of phospholipids, and you can see the way the 2 layers are arranged. 2 layers of phospholipids are arranged with their tails together, as if their feet or tails are together and their heads are apart. And the reason you'll come across this later on, the reason they're arranged like that is that the heads on inside the cell are in contact with the cytoplasm, which contains a lot of water, and the heads on the outside are in contact with the what you call the tissue fluid outside the cell, and it also contains a lot of water. And the tails are protected from being in contact with water because the tails are pushed together. The 2 tails are sitting together in the middle, and so that you will do the cell membrane in a separate topic, and you'll see the detail of it. So that's a very simple diagram at this stage. Any question sometimes get asked then is compare a lipid and a phospholipid. It may ask you compare a triglyceride and a phospholipid, but basically it's a lipid and a phospholipid. You can see there the lipid has 3 fatty acids, phospholipid just has 2. The lipid has no phosphate, no P group, no phosphate group, whereas the phospholipid does. And then you would also say that one of the properties of the lipid is that it's not hydrophobic. So the lipid doesn't have these 2 ends that are either attracted or repelled from water, whereas the phospholipid does. It has a hydrophilic end and a hydrophobic end, hydrophilic head and a hydrophobic tail. So got as far as lipids, and probably about an hour has elapsed there. I'm just noticing. So it's quite a long topic, this. So next time I'll I'll try and make a short video maybe for on proteins. So for now, I'll leave you with the lipids, and we'll stop there. And okay then. Thank you very much. Bye.